microscopy, and I'd like to welcome you to this installment of our webinar series. Our presentation today is titled Live Cell Super Resolution Imaging, and it's being presented by Dr. Manasa Gudheti. And before I introduce Manasa, I'd like to just make a couple of quick logistical comments. First of all, we encourage your participation during the webinar, and if you have a question, just type it into the questions dialog box that's on your screen. Those accumulate throughout the presentation, and at the end of the presentation we'll work through the questions. Occasionally we have more questions than we can answer in the time allotted for the session. If we have cases like that, we'll make sure to follow up with you personally with an email afterwards. Also, if you'd like to review the presentation afterwards or pass it on to a colleague, this webinar is going to be posted online within a week in the webinar section of the Bruker webpage. And as a follow-up to this presentation, you'll receive an email with a link which you're welcome to share with your colleagues. And then finally, when you exit this webinar today, the software is going to ask you to take a brief survey. We'd really appreciate it if you could take a couple of minutes and complete that, because it really helps us refine the topics that we pick, and most important to you, and generally to make this series as beneficial as possible to our scientific colleagues. So let's get started. Let me introduce our presenter, Manasa Gudheti. Manasa earned her Bachelor's in Technology from National Institute of Technology, Chemical Engineering, followed by a Ph.D. from Drexel University in Chemical Engineering. She worked in Sam Hesse's lab at the University of Maine for her postdoc in biophysics. This is the lab where the F-palm technique was developed. She's been in the super resolution field now for over 8 years, and she's a great scientist, and she's definitely an advocate for the field. So with no further ado, let me now turn it over to Manasa for her talk entitled Live Cell Super Resolution Imaging.
Thank you, Jeff. Thank you for the introduction, and good morning. Welcome, everyone. So today I want to give you a brief overview of the advancements in live cell super resolution imaging, specifically in the single molecule field. This is an area that is rapidly evolving, and compared to doing fixed cell imaging, there's a lot more factors to take into consideration, and I want to go through each one of them and kind of give you examples of how to optimize those factors so that you could get the best super resolution images possible and thus drive your science forward. So in terms of the outline, just want to give you a very brief overview of super resolution microscopy, why it's become so popular in the last decade or so, and talk a little bit about single molecule localization. That's the technique that the Vutara 350 microscope uses, and we could do 3D super resolution imaging along with really high-speed imaging, and I'll talk about the technology in great detail, what makes this technology unique and how it's ready to be able to be harnessed for live cell imaging. And the factors crucial for successful live cell imaging. There are several factors to consider before you embark on this, on doing these experiments, and the more prepared and aware you are, the more successful you will be in that sense. And I'll talk in detail about the Vutara 350 microscope, the hardware, the software, and the different aspects that come together to make it live cell ready. There's a lot of sample prep optimization, the type of dyes you want to pick, how you want to do the imaging. There's a lot of stuff to take into consideration when you're doing live cell imaging. So I'll talk about that as well. And then finally, what is driving this field forward, and in order for this to become very standard, so that a lot of people can use this technique, especially for live cell experiments, is new dye development. And there are several labs around the world who are driving this, and then there are companies that are commercializing these dyes so that scientists have access to a lot of labeling techniques to be able to use for their biological system of interest. So here's like a brief overview of super resolution imaging and why it's become so popular in recent years. So in the past, you had two options essentially if you wanted to do microscopy. If you wanted to look at live cell imaging, 3D, and all that stuff, you would essentially either do wide field or confocal microscopy. And if you wanted higher resolution, then you would choose EM. And each one has its advantages and disadvantages. one of the main advantages of being able to do light microscopy is that it's non-invasive and you're able to do live cell imaging with that. So in order to kind of combine the best of both worlds, where you're getting the enhanced resolution with DM but still being able to look at live biological samples, three main super resolution techniques evolved, and they're always compared against confocal, especially in terms of the resolution that you can get. So the first technique was structured illumination, and you could get about a twofold improvement in resolution there in X, Y, and Z. And for some applications that works great. One of the advantages of SIM is that it uses a lot of the traditional fluorophores that you're using for confocal, things that are photo bleaching resistant, and you can kind of use that for SIM, and that's been a big advantage of that. But it's still a twofold improvement in resolution. So with STED, you can essentially get about fivefold improvement in resolution in X, Y, and Z, just depending on what flavor of STED you're using. So you can definitely get the enhancement in resolution, but also requires specialized dyes that are very photo bleaching resistant, and there's been a lot of development going on there. So the Vutara 350 microscope is based on single molecule localization methods. So that will give you a 10-fold improvement in resolution. So you can get about 20 to 30 nanometers in XY and 50 to 80 nanometers in Z. So this includes the wide array of techniques, PALM, STORM, D-STORM, GSM, whatever you want to call it. But what we essentially call it is single molecule localization, where you're essentially looking at the centroids of the molecules and building up an image over a period of time. So this technique offers the best resolution, but there are some sample preparation optimization that you would have to do to get the best out of this, and I'll talk more about that. So in terms of how does the technique work, the concept itself is fairly simple, where you're labeling your sample. You're either photo switching or photo activating the molecules so that you only have a few on at a time, and then you're essentially building up the image, as you can see here. So what's unique to the Vutara 350 microscope is that it's actually doing this in 3D. So we are collecting from two simultaneous focal planes, which are separated by 500 nanometers. So even without having to do a Z-stack, you can get information from about a 1 micron depth of field of view. And if you wanted to image thicker samples, then you can go ahead and do Z-stacks. So in order for this blinking mechanism and everything to work, there are certain dyes that work better than others, and I'll give you examples of those. And then when you start thinking about live cell imaging, you also have to start thinking about, okay, what type of powers do I need to use for the switching to happen? What kind of dyes can I use? What kind of blinking buffer do I use? So those are some of the considerations that you have to think about when you're doing live cell imaging, which you really don't have to worry about when you're looking at fixed specimens. Okay, so this is the key thing. These are the factors to kind of keep in mind if you wanted to do successful live cell single molecule super resolution imaging. So the first thing that needs to happen is that you need to have the technology that can make this happen, both in terms of the hardware and the software. And the first consideration, of course, is you need to be able to have flexibility in terms of how fast you can image. So the speed of imaging becomes really important in live cell imaging, so that you can capture the dynamics at the right temporal scale. Along with that, you should be able to match your dye photo switching kinetics with the temporal scale that you're looking at. So if you want to image really fast and looking at fast dynamics in a biological system, then you need to be able to have high-powered lasers to actually drive your photo switching kinetics. And I'll go more in detail about that. And in terms of the software, you need to be able to have very fast, pretty much real-time localization of molecules. So as you're acquiring, you need to be able to localize the molecules. And this is not a trivial matter, especially when you're looking at something in 3D. So you need to be able to localize these molecules in 3D in pretty much real time so that you can decide what the next course of action will be. And the backbone of all of this is the sample prep optimization. In order to get the best spatial and temporal resolution, you need to pick a dye or a fluorescent probe that is compatible with the system that you're looking at, that can blink, whether in the buffer that you want it to blink or without buffer, or with the minimal amount of blinking buffer possible, whatever works for the experiment that you're planning. And finally, you want to choose, again, a dye that's compatible with live cell imaging. So especially when you start thinking about live cell now, labeling external, like surface proteins, is a lot easier because you have a lot more options to do that than something inside the cell. So now we have a choice of cell permeable fluorophores and ones that actually switch without switching buffer, so you don't have to worry about permeability of buffer into the cells or toxicity. So these are all the factors that you kind of have to keep in mind when you're designing a successful live cell experiment. Okay, so first I'll talk about the technology aspect of it. So you need to have a super resolution microscope that's going to give you the resolution that you're looking for and be able to image at the speeds that you're looking at. So here's an image of the Vutara 350 super resolution microscope. So it is the fastest super resolution microscope on the market. We could do video rate imaging. We use an SCMOS camera in order to be able to achieve that, so we can image up to 3,000 frames per second. We have high-powered lasers in the system in order to be able to drive the fast photo switching kinetics. And the resolutions that we are able to achieve are about 20 nanometers in XY and 50 nanometers in Z. There is easy to use, a very powerful software which harnesses a lot of the computational powers that's available now to be able to do real-time localization. And one of the crucial features of our software is that you could do multi-emitter fittings. So if you're doing really fast imaging, the chances that their molecules that are the PSFs are going to overlap starts getting higher, and you need to be able to account for that so that you can actually image faster and still be able to get a very good super resolution image. So let's talk about how we're using the SCMOS camera in order to achieve this high-speed imaging. So traditionally, ECCD cameras have been used for single molecule localization applications. So kind of the comparison between an SCMOS camera, we use the Hamamatsu Arca Flash 4, just comparing it with the standard ECCD camera. So one of the big advantages of using an SCMOS camera, of course, is you have a bigger field of view, 2048 pixels versus 512 by 512 pixels. And then the frame rates that you can go are also considerably faster. At full frame, you can kind of go 100 frames per second on the SCMOS, whereas it's about 56 frames per second on the ECCD. And in terms of quantum efficiency, it's actually and I'll show you a graph in the next slide, where the quantum efficiency, the effective quantum efficiency, is actually higher on an SCMOS camera than it is on an ECCD camera. So one of the biggest factors to consider when using SCMOS cameras is that the readout noise is actually pixel dependent, and that's something you have to be able to account for if you want to use SCMOS cameras for single molecule applications. So that's what we call our res-enhanced technology, which was published, and we have it patented, published in Nature Medicine last year. So as you can see, there's a pixel-to-pixel variability in the noise characteristics. So if you wanted to do single molecule applications, that would be a barrier. And this is, people have been aware of this for a while, but in order to account for that and use it in your fitting algorithms for localization, you needed to have a very computationally effective way of doing that. So that's what we essentially did. We adapted that. So now when we're doing the localizations, we take into account these the pixel map characteristics here, and then that way, applying the rest enhanced technology, you're able to use the SCMOS camera for doing single molecule localization applications. Okay, so one of the questions that we get is, well, how does the SCMOS camera compare in terms of EMCCDs, in terms of the number of photons, how sensitive it is? So as you can see from the graph here, so this is the incident number of photons, and then you're looking at background. So in situations where you have extremely low background and extremely low light levels, the EMCCD yields better performance. But for the most common experimental conditions that you're looking at, the SCMOS camera actually performs better. So that's a factor that you don't have to worry about. In most cases, you're kind of at least looking at 100 photons per pixel or higher. So basically, if it's less than 6 photons per pixel, then the EMCCD will work better. But for most other applications, you can use the SCMOS camera and get better performance. Of course, you have a larger field of view and you can image faster. So it's an ideal camera to use for live cell super resolution imaging. So what are the kinds of speeds that we are talking about here? So these are examples in fixed cells, but just to give you an idea of how fast can you acquire an image. So here's an example where you're taking 800 frames per second. The total number of frames taken were 2,000. So about 2.5 seconds, you're able to build up this one-color image. So we're looking at Alexa 647 labeled alpha tubulin. We can do two-color imaging. Again, 800 frames per second and 2,000 frames. Up to 800 frames per second, you can kind of get this 200 by 200 or up to 400 by 400 pixel area on the SCMOS camera with the Vutara 350 system. With the reduced ROI, you can image up to 3,000 frames per second. So here's an example of imaging at 2,500 frames per second. So this is the area where if you're looking at really fast dynamics, those are about the speeds that you'll start going at. And in about 4 seconds and then 10,000 frames, you're able to build up an image. So as long as you're able to match the labeling density, the powers, and then the acquisition times, as long as you can optimize that, you should be able to get really good images on seconds and sometimes minutes time frames, depending on how thick of a sample you're looking at. So in terms of high-powered lasers and how do you control the switching kinetics, so if you're going to image faster, you also need to be able to switch your molecules faster. So typically on an EMCCD camera, you might image it at 50 to 60 frames per second, and then you can use low powers so that you match the switching kinetics to the acquisition speed of the camera. But then now in the case of an SCMOS, since you can go really faster, you need to be able to get your molecule to go from an excited state, and back to the ground state in a faster time. What Zhao Zhuang's lab did was characterize the switching rate constant as a function of laser intensity and how that corresponds to the number of photons that are emitted per switching cycle, as you can see here. So what they found was that as you increase the laser power, you can drive the kinetics to go faster while still keeping the number of photons per switching cycle the same. So which is great news, which means if you wanted to image at, instead of 50 frames per second, you wanted to image it at 200 frames per second, now you can use a higher laser power, still keep your number of photons the same, so you're keeping your resolution the same, so essentially you have a really good signal-to-noise ratio. And so the faster you want to go, the more laser power you need. So which is why we offer up to 1 watt lasers in 4 different colors, and the 405 comes up to 100 milliwatts so that you can do photo activation as well. And so this actually ends up being really crucial for doing live cell imaging, is having access to high-powered lasers. So when you're also imaging at such high speeds, like I said, at lower speeds, you're going to get a scenario where most of your molecules are actually separated. So there's essentially one or two molecules within a diffraction limited space, so you can clearly separate out their point spread functions. But when you start imaging really fast, the scenario is bound to happen. So then you need to be able to account and fit for those overlaps using a different set of algorithms, which are incorporated into the software when you're imaging faster. So that's the software end of things. So how do we do all this? How do you do the multi-emitter fitting and real-time localization? So we really harness the power of parallel processing. So a highly parallel architecture. So we use the GPU and also the multi-threaded CPU, a multi-core computer, to be able to do real-time localizations. So we have a scalable architecture, so you can do up to 40 terabytes. So this essentially enables us real-time localization as we're acquiring the data, and also be able to kind of jump from localization, acquisition to localization to visualization, and also be able to implement multi-emitter fitting because it is very computationally intense. So you'd be able to use the computational power that you have to be able to do real-time localization. Okay, so that was in summary sort of the hardware and the software that you need to have in order to be able to do live cell imaging. So in terms of now switching gears, and in terms of what can we do at the sample. So in terms of sample labeling choices for single molecule localization is you've got two options. Basically, people either use organic dyes or genetically encoded fluorescent proteins. And typically, organic dyes are preferred, especially when people are starting out for SML labeling over fluorescent proteins. A couple of reasons. One of the biggest reasons is, of course, they emit about 10-fold number of photons than the fluorescent proteins do. Two, if you're looking at typically fixed cell applications, you could just do immunolabeling, and you don't have to put in a lot of time and effort up front while you're trying to test what this can do for genetic encoding. But on the other hand, fluorescent proteins are more physiologically relevant, then they're more live cell compatible. So there's kind of advantages to both, and so people have been picking whatever works for them in order to be able to do the experiment that they want to do. So in terms of the preferred dyes, I mean, I've tested a lot of the dyes that are available out there. You've got a couple of them in each wavelength range that work really well. So for the 488, the Auto 488 works really well. For the 561, the Psi 3B and the Alexa 568 work well. For the 640, the Alexa 647 Sci-Fi work well. And then for the 750, the Alexa 750, and then the Dialite 755 work really well. So you've got these range of dyes, but of course when you start thinking about live cell applications, a lot of these can be easily adapted to doing surface labeling. But once you want to start doing internal labeling, what options do you have? And I'll talk about technologies that have made it possible to kind of combine the best of both worlds there. So in terms of photo-switchable proteins, you've got a variety of options, but in terms of the ones that work really well, MEOS 3.2 and then Dendro work really well. The rest of them are okay, but they're nowhere close in terms of the photophysical properties. So you can do really good live cell experiments, genetically encoding and all that stuff, but then when it comes to multicolor imaging, the fluorescent proteins you start having limitations. You want to extend to 3-color, 4-color. You do start running into a few issues there. But if you're kind of doing 1 or 2 colors, and you can optimize it. So here's an example looking at the diffusion of the flu protein on the surface of actin. So looking at Dendro2 HA and PM cherry actin. So this was imaged at like 50 frames per second. So the number of photons that Dendro-2 was emitting was essentially enough to be able to capture these dynamics, and the lab was also be able to extend this to 3-color imaging, where we found you can kind of do this, but there is bound to be spectral overlap, and the Vutara 350 microscope will be able to, since we can do 2-color simultaneous imaging, you will be able to do ratiometric imaging, but it's definitely a lot trickier to do that than using organic dyes that are very, very spectrally separated. But so you've got some options in terms of doing multicolor imaging here, and here's a video looking at the diffusion of transferrin and hemagglutinin on actin again, and then you can see that the transferrin forms these linear tracks. So essentially you're plotting this every 2 seconds, so you're getting about a temporal resolution of 2 seconds here, but the fluorescent proteins that you're using. So here's another live cell imaging example of MEOS 3.2 clathrin light chain, done by Jörg Weberstör's lab at Yale. So this was basically imaged at 600 frames per second, so that's your raw camera speed. It was imaged for a total time of 58 seconds, and then if you plot all that data at once, you can see it's kind of blurred, so you're actually missing out the dynamics. But if you separate them out into 2-second intervals, you can essentially see the diffusion of these clathrin-coated pits. So you're able to image really fast and then pull the data to be able to pick the time scale at which your dynamics are happening. So let's suppose you wanted to look at dynamics which are even faster. So in that case, you wouldn't have, MEOS 3.2 wouldn't give you enough number of photons in order to be able to image on the system. So you need to turn to organic dyes. So in this case, it was surface labeled. So this is Alexa 647 labeled transparent. It was imaged at 1600 frames per second, and then essentially 50 frames of data. So 37 milliseconds worth of data was acquired. So that's the temporal resolution that we're getting. So in this case, the switching buffer was optimized such that it was live cell compatible, where you're essentially imaging it in phenol red media and using very little amounts of tile so that it doesn't affect the live cell characteristics. And then it was imaged at room temperature here. So here's another example of just looking at a track of transparent molecules. This was imaged at a slower speed, but then you were able to essentially get a trajectory of how the transferrin molecules are moving. So it just kind of gives you an idea of the different types of experiments that you can do. So people have been using the organic dyes and then the fluorescent proteins to do live cell imaging. There's a few other examples that I want to share with you because there are some membrane dyes and structural dyes that also can be adapted for doing live cell imaging. So here's one example from Suliana Manley's lab at EPFL, where they did live cell imaging of direct labeled DNA. So this dye is pico green, and they essentially just incubated it into the cell, and as you can see, they're not using any thiols. They're actually using ascorbic acid to be able to do the switching, and they were imaging it in glucose oxidase and catalase and be able to get the dynamics of the cell. So this is essentially a conventional image. This is one frame of where you can see the switching molecules, and this is the built-up super resolution image. And then this is, of course, was imaged live. Another example of super resolution imaging in organelles. This is from Xiaoyu Zhuang's lab, published in PNAS in 2012, where they were able to use some of the standard dyes that people use, like dye for plasma membrane. So here's an example in a hippocampal neuron here. They also imaged mitochondria with mitotracker red in this image, ER with ER tracker red, and then lysosomes with lysotracker red. So The one caveat with all this is that you just want to make sure that you're getting, you're just using the right incubation times, and then you're also optimizing for low background. So whatever you can do to minimize background and up your signal will definitely make a big difference in the quality of image that you're going to get. Here's an example from Mohrner's lab, W. Mohrner's lab, the one who, one of the Nobel Prize winners for single molecule localization. So they were one of the pioneers in using photo switchable. So they could photo switch EYFP, enhanced yellow fluorescent protein, looking at this in bacteria and imaging it live. So they found that you can kind of use it as a live cell organic dye sort of way, even though it's fluorescent protein. And they were able to do a lot of experiments in the Caulobacter crescentus cells, and they've published extensively. If somebody's in the bacterial imaging field, I would encourage them to check this. Mohrner's publications. There's a lot that they've done with this type of biological system. And in terms of being able to image without switching buffer, there are some dyes out there that have been engineered such that they can be imaged in PBS, and a lot of development is going on in that field as well. So here's an example. This is in a fixed cell, but looking at KH505 labeling clathrin, which is in green, and then 565 dye. So basically, you can image them at lower laser powers. I know the KH505 is actually cell permeable. I'm not sure about the 565 dye here, but then yeah, I was just able to prep the sample and then image it in PBS. So you can kind of start thinking about, oh, okay, I don't have to use switching buffer. I can use low laser powers. So, like phototoxicity things can also be controlled in that fashion. So now, as I said, proteins versus organic dyes. So how do we combine the best of both worlds, which is essentially what we want? And a lot of folks have thought about that. So in the last few years, new labeling technologies have been developed to exploit the best of both worlds. So the most common ones that people use are snap, clip, and halo tags. Snap and clip tags are commercially sold by NEB, and then Promega sells the halo tags. And the basic concept for all three of them is very, very similar. So essentially, you end up getting a snap tag, clip, or a halo tag vector, and then you would tag, you'd essentially create a clone, and then you tag your protein of interest. You tag the snap tag to your protein of interest, as you can see here. And it's got a motif here that it recognizes. And basically, you would transfect them or electroporate them into your cells, and then you would incubate with the ligand that you're interested in. So basically, the label that you're interested in. So, and I'll show you a list of different dyes that are available for this. But it's basically just incubation, and then you wash it off, and then, as you can see with the SnapTag fusion protein, it just tags on to the its substrate here, releases a guanine. In case of a clip tag, it releases a cytosine. In terms of halo tag, very similar phenomena here. It's also an enzyme-based type of technology. So in this case, since they all have different motifs that they recognize, you can kind of combine to do 2-3 color experiments. So it's very specific, very fast, very reliable. So you're essentially doing transfections or electroporation, but at the same time, there's a lot of dyes that are cell permeable, and since you can live label them, you have great flexibility for doing live cell imaging and still be able to get the high number of photons that you'd get for organic dyes. So this is the field that is rapidly evolving now in terms of doing live cell super resolution imaging. So So for anybody who's interested, the snap and clip tags came out of Kai Johnson's lab at EPFL. So these are the initial publications, and then this is a publication for the halo tag. So as you can see, they've been around for a while, but in terms of just being able to engineer it in such a fashion that they can be used for super resolution imaging has been happening in the past, 4 to 5 years or so. So there's a variety of snap, clip, and halo tags. So the greatest variety you're going to get is with the snap tags from NEB. So you've got the snaps, the cell dyes here are all cell permeable. So you can see you've got 505, TMR star, silicone rhodamine dye, which is something that recently came out of Kai Johnson's lab. And I've had the chance to work with one of the postdocs in Eric Jorgensen's lab here at the University of Utah. And TMR star and SIR are actually really great dyes to be able to use for this. And then, of course, there's clip. The clip doesn't quite work as well as snap, but it does end up giving you a second color. So we've typically been combining snap and halo for doing two-color imaging, and I'll show you a couple of examples of that. So I want to give you some examples of in literature where people have used these snap and clip technologies essentially to do live cell imaging. So one of the first publications to come out was from Xiaowei Zhuang's lab, where she was using a Surface Alexa 647 snap tag, directly labeling transparent with Alexa 568. So the Clathrin-coated pits are labeled here in magenta with the snap tag, and then as you can see, this was a live cell experiment where they could actually see the movement of the Clathrin-coated pit. This is from Marcus Sauer's lab, the one who developed D-Storm, where they're looking at histone, looking at snap cell TMR star. So as I said before, TMR star is actually cell permeable, so you can look at nuclear proteins. So in this case, they were actually, they used the snap cell TMR star to be able to build up the histone proteins in a living cell. So the snap and clip technologies and halo technologies have been around. There's several dyes that are available. A lot of them do need to have some kind of switching buffer in them. There's some that don't, and then there are folks who are working on developing technologies, dye technologies, so that you can actually have spontaneous blinking with just optical control rather than chemical, putting anything chemical. So this is a fairly recent publication in Nature Chemistry where they essentially modified rhodamine derivatives to be able to generate a spontaneously blinking fluorophores. And then I believe they were able to, I encourage you to check out the publication here, but they were able to image for about an hour looking at tubulin. But the nice thing is, this is kind of a comparison between what happens if you're using very traditional, like Alexa 647. You would need to use a higher laser intensity, and you need to have some kind of reducing agent in your buffer. So in this case, you would need to use additives, and it's a lower laser intensity. This is something, of course, you need to test out for your own system, but there's a lot more choices that are being available now. In a similar vein, this is a SNAP silicon rhodamine, which was published last year, and then now the SNAP version is actually available, commercially available from NEB. So this one, you can do imaging in just labor with this medium. That's what they ended up doing the imaging here. So you can reconstruct the images in something that doesn't have switching buffer. And also, I've worked with the SIR, or the silicon rhodamine dye, and then essentially it switches at low laser powers than the Alexa 647 would switch. So I kind of want to give you two examples of how Eric Jorgensen's lab here in conjunction with one of the postdocs, Rob. They've actually used live labeling. These are fixed worms, but they're actually live labeled. So we're just trying to work out conditions where we can do live worm imaging, essentially. So the green is a snap tag with TMR star, and then the red is a endosomal marker with halo tag. So as you can see, you know, you could do live labeling and get really high resolutions. So you're essentially looking at the ventral cord here, or you're looking at the cholinergic and GABAergic synapses. So of course, you need super res imaging to be able to get this high level of detail. But if there are any worm people listening, you know that it's, you know, labeling, antibody labeling in worms is, you know, really, really hard. So the SNAP and CLIP and halo technologies have made it possible to do live labeling and still be able to use permeable or membrane permeable or cuticle permeable dyes to be able to do super resolution imaging. So here's another example looking at the NMJ's neuromuscular synapses here in the worm. So this is one of my favorite images, so you can kind of see the active zone marker and the synaptic vesicles that you're marking here. So yeah, in summary, what I want to say is that, you know, you need to have really good hardware. So in order to be able to a really good microscope, essentially, that's been optimized for live cell imaging hardware and software-wise. Sample prep optimization in terms of choosing the right dyes, choosing the right imaging buffers, all those things have to be paid attention. And you know, the development of new dyes and labeling techniques is what's going to propel this field forward. And I've been in this field for a while, and you know, in terms of, I think, live cell imaging, we're finally coming into that era where there's going to be a lot of exciting developments going on. So my idea with this webinar was to present you with just sort of like a bird's eye view of what's possible, what's out there, so you can start exploring different options and sort of have an idea of, you know, what needs to be considered if you want to embark on this, you know, I think, an exciting journey. So with that, thank you so much for your attention, and I will be happy to answer any questions. Thank you.
Okay, thanks, Manasa. Great talk. I have a few questions here. First question is, one of the listeners, using such high power of lasers will cause phototoxicity to live cells. How can you minimize such damage on live cells? That's
a very good question, Jeff. So one of the things that I always point out is that this is a wide field technique. I mean, you're using high-powered lasers, but you're actually spreading out the high-powered the laser power over a larger area. I mean, you're looking at like a 20 by 20 or 40 by 40 micron area. All this laser power is not concentrated in just one pixel. So that in itself is a great thing because you're minimizing it that way. And in terms of just when you compare confocal versus this, you're actually per pixel you're putting in way less power than you would. But having said that, the goal in terms of optimizing this is pick dyes that will use low laser powers and will switch at low laser powers. So in essence, pick far red dyes rather than doing green dyes because green dyes take more laser power to switch. Excuse me. So you can minimize that, and of course, also remember that the time frame of your experiment is fairly short. If you're imaging really, really quickly, and then minimizing the amount of exposure that you're getting, that will also minimize phototoxicity. And you just have to use just enough to keep your cells happy and not kill them, and still be able to get good results. So it's kind of that delicate balance.
Okay, great. Another question. How do you determine the temporal resolution in a live cell single molecule localization experiment?
That's also a very good question because, like as I spoke, I mean, if you're like a physicist and looking for, oh yeah, this is I can go 500 frames per second, 800 frames per second, 3,000 frames per second, and I think for a scientist doing biological experiments, that doesn't mean much because, ultimately you're going to say, okay, what's the time resolution that it takes to be able to get the information that I need to get? So essentially, you want to see how much you have to pull your data. So in case of the collateral-encoded pits, even though we were imaging at 600 frames per second, the temporal resolution was effectively 2 seconds because we had to pull 2 seconds worth of data to be able to get the dynamics that we were looking at. And for some applications, that works really great. And in some cases, you might need less amount of data to be able to do that, like in case of transfer, and it was only like 31 milliseconds. So that's something to kind of keep in mind. It's great to be able to go really fast, and that is essential, but matching your dynamics, temporal dynamics that you're looking at in your system with the acquisition time is what is going to be finally determining how good of an image you're going to get.
Okay. How deep into cells or tissues can you go?
Okay. So in terms of the hardware itself, I mean, the piezo has about a 400 micron range, and then we've got, we use the water objective, so it's one of the nice things. So you can actually image deeper, so that's about a 270 microns depth of field of view that you can get, like depth that you can actually go into. So essentially, after that, it just depends on the sample and how do you minimize scattering. And in live cell imaging, that might be a little bit more tricky. With fixed cells, you can kind of use clearing agents. So if you can somehow minimize the background in live cell, live tissue or live cell, whatever you want to look at, then yeah, about 270 microns would be the physical limit.
Okay. So another question, are there any good genetically encoded labels?
Yes. So the best ones that I have seen so far have been MUS 3.2 and then Dendra 2, and then to a certain extent PM cherry actin work really well. I personally worked with all three of them, and they're great. If you want to go the SNAP and CLIP and HALO route to be able to do genetic encoding and still be able to use organic dyes for high-powered imaging, then I would say TMR star is great. We worked extensively with that. For surface labeling, Alexa 647 works really well. And then the silicon rhodamine dye, has actually been really great. So we've tested that out in the lab here extensively. So those are the ones that I would look at initially if you wanted to kind of explore things.
Kind of along the same lines, what probes, either organic or fluorescent proteins, are currently the most optimal for live cell super resolution imaging?
Again, yeah, if I had to pick the best, I think for most people MUS 3.2 would be it. And then if I'm doing surface labeling, Alexa 647, that's what I would pick. And if I was doing intercellular labeling, probably like a TMR star or a Cage 500 type of dye would work really well because they're all live cell compatible.
Okay. And last question we have at this point anyway is, how would you go about optimizing sample prep and imaging conditions?
Yeah, that's a great question. So I think initially I would start off with, what am I, what is the temporal resolution that I'm trying to achieve? Could I get away with just using an MUS 3.2 type of thing? If I'm looking for, dynamics on the scale of 1 to 2 seconds, I think those work out really well. And if I want one color, that's what I would go with because the imaging is going to be much more straightforward. But then if you want to do 2-color imaging and you want to be as physiologically relevant as possible, then I think, yeah, I would kind of do like a SNAP and a HALO combination. And that would involve a little bit more effort, but then you're going to get highly specific, genetically encoded system. But then, yeah, you can then test out different dyes. Is it also 647? TMR7 or TMR star that works for me, you'd be able to live label those. And just essentially, most of them they switch the glucose oxidase catalase even without any diols in them. So that would kind of be my first step for optimization. And after that, I'll just see, do I need to minimize the background? Do I need to up the amount of dye that I want to put in there? How fast can I go? If I go too fast, how does my signal to noise look, and things like that. So that's kind of my thought process when I'm designing a live cell experiment.
Okay, great. No more questions at this point. Certainly, if you have any questions come to mind, you can go ahead and email those in, and we'll get back to you on that. And again, first of all, thank you everybody for your attending today, and I'd like to remind everybody it would help us out a lot if you could just take a quick survey when you exit the webinar application. Thanks, Manasa. Great talk, and thanks for everybody attending today.
Thanks, Jeff.